So that so that's kind of what the initial pilot data and what the idea is. So kind of next steps, we do need funding to secure more actigraphs. We did also want, if it's going to be multi-centers, pay for coordinator time, maybe provide patients gift cards to incentivize them if they were to wear it for 20 hours a day for 7 days at a time during the study period. What is the best funding source? Obviously, apply a grant submission to the NIH one, and if it is accepted, will be something which is really good. And then obviously ideas, suggestions, and how to improve or kind of tweak our study design since it's still in the developmental stages. So that's what I've got.
All right, very interesting. And it's really nice to have these questions to frame parts of the discussion. I think it's a really interesting study you've done and really intriguing. So I'll open it up to discussion. So please feel free to unmute yourself and ask a question or put some questions in the chat as well if you wish.
I have a question. So is there any possibility for collaborate with the basic scientists on your project?
We definitely would love to collaborate, as in what that might look like or how that plays into the study design. I'm not sure, but absolutely, we're always happy to collaborate and definitely build on what we've got.
What kind of thoughts do you have about a potential collaboration, Yaping?
I have no idea. So I think about, what kind is there any animal model to be like a mimic?
Okay. I'm just kind of making notes, but yeah,
some kind of.
Go ahead.
No, this is Gary. I was just, mentioning that some kind of pathophysiology piece to it with movement and the correlation with lung capacity, something of that sort. Maybe study that in animals and go from there. Maybe I'm just trying to think out of the box here from based on, I don't know how we'll measure animal activity and things like that, but that's the only thing I can think of and try to see the changes in pathophys as such. Because, I know you mentioned some subgroups of patients where, they have either heart failure or some other kind of infectious or, cancer etiology causing the perfusions. Is there a difference? And is there a clear correlation between activity after and before you do the pleural tap and things like that? So that'll be the only thing I can think of. But it's a great study. I think from your questions though, are there any other studies like this for lung capacity in general? Not necessarily. Sorry, I just started and followed you a little bit later in, but not just necessarily pleural effusion, but other kind of lung disorders and using pedometers or any kind of physiology monitoring.
Yeah. So these have been validated actually in other disease states like COPD and cystic fibrosis. And there's one small study in pleural effusions too. So yeah, this has been actually validated in other disease states. A lot of the studies have kind of focused on nothing which we've done over such a long period in time, like 30 days, wherein they've all focused on like 6-minute walk tests and gait speed and things like that. So the active is fairly validated.
So for funding, did you see any of these getting big R03s or big kind of
I
big
have
funding?
not, as in
fundings?
the only out of three examples I looked at is what's already on the NIH website, and one of it was to do with molecular analysis of histoplasma, and then there's another one about some atomic subgrouping. So they just had a wide range of examples on it. It doesn't, when I look through it, it doesn't kind of rule anything out or rule it in. It gives you just instructions on how you need to put together your proposal.
So we have a lot of faculty on this call that have had substantial amounts of NIH funding. I think it would be good, even if this is not your field, if you have thoughts about, this is preliminary data for the kind of studies proposing a larger one. Being able to separate different populations based on disease, et cetera, I guess. Whether that's a good fit, I don't know. I'm really interested what other people say. I think usually for NIH, having some kind of mechanistic or pathophysiologic information in addition to just an output is usually, I don't know, usually that makes it more appealing to reviewers. That I think your goals, when we've talked previously, are do a larger study, find out maybe if, even though patients don't subjectively think that they have a lot of improvement with thoracentesis, maybe their activity levels are higher, and that you have a window of time for physical therapy to even further increase, the benefit of it, because they're more capable of participating in that window of time. I don't know, what do some others think?
Yeah, I feel like typically, even though this is a pure clinical study, but the review panel typically have 1/3 as from basic science. So they like some kind of mechanistic study. So you have if it's purely those outcomes, so they will view this is more like an observation. So like to have your initial observation data point out this is a really clinical significance, and there's it's a feasible, and then you have some kind of a mechanistic study involving, and then see the improvement, and even have some kind of intervention to improve that. There'll be, I think that this is a very good initial study to show that it's a promising, but adding some model system. That's why I see whether the basic science, if there's a model system available, so they can use the same strategy applied to the model system, and then do some kind of therapeutic or whatever, some kind of improvement that they'll make the ground application much more stronger.
So are there any studies from our PTOT schools that get NIH grants for kinetics, and how are they connected to basic sciences, or how do they obtain their grants? Maybe that's another thought just to kind of explore. I mean, because this is more of kinetic in a way, if you really think about it, and how the pathophys with I'm actually talking a little bit out of my comfort zone there, but in terms of oxygenation of tissues and things like that, and how that translate into mechanical work, that way is more objective kind of thing. So just wondering how they get their grants, because they don't have.
I can jump in on that too, because if I'm understanding this correctly, NIH funding, then they seem to not fund clinical outcome only projects. Is that, that basically the bend that I'm hearing here, or is it because the grants that folks are getting here are generally coming from basic science? We had a meeting. So I was involved in this project too, but we had a meeting, I don't know, 2 or 3 weeks ago with an individual from Mayo who kind of pushed Nikhil towards, pursuing NIH based on these results. And this individual has millions of dollars in NIH grants as well, and he's a clinician. So maybe the question back to him might be, do you always have to have basic science, or is it possible, or are we thinking no NIH right now for where this project stands, and maybe more foundation funding to get more types of data, et cetera? Because again, the reality is we don't want NIH or lack of NIH funding to be the reason that this stops, right? But we definitely need funding to make sure that we can answer these subsequent questions. So finding kind of that best avenue is where we're at right now, and that's where we want your insights.
So from reviewing in a lot of NIH study sections focused on cancer, for example, many applications are submitted by clinician scientists, and many clinical trials are, the heart or the central part of those applications. But I think they're filled in a little bit. It's not just, this clinical outcome, yes or no, but there's, there might be samples taken for some molecular analysis to, I do mostly cancer stuff, or developmental biology. So it might be, look at these samples to see whether the target is being actively engaged by a drug or whatever. So there's some additional aspects to it. I think going to an animal model would be a big step backwards, really. I think that would be a huge leap. And, are the animals going to behave the way people do? I kind of think if there's a way to, I don't know, maybe to have additional endpoints come out more than just activity and reported symptoms. I don't know could you have lung function, a more, an objective measurement of, I don't know, of some kind of lung function or a little bit more of that to give people a little bit more to put their teeth into.
So from a clinical standpoint, and that's kind of why subjective symptoms have always been there and will be around, is at the end of the day, if a patient comes and says, I feel breathless, that procedure will be done if the fluid is there and it's indicated clinically at that point in time. Because we're not going to withhold a procedure saying that you did not do much in terms of activity, or activity did not improve. So the breathlessness or the shortness of breath, those subjective symptoms are going to be there. And then looking at it from just a pure clinical standpoint is, at the end of the day, for me, if somebody says, yes, I feel better from a symptomatic standpoint, I feel like my activity is better, then their labs or their lung function or their overt walk distance is not, it doesn't matter.
Mm-hmm.
So removing that extra 510 cc of fluid is not going to change. That wouldn't change. So from a purely clinical standpoint, if there's a clinician on the panel, is what is the lung function going to matter? So even if somebody's forced vital capacity improves, but overall they don't feel any different in terms of subjectiveness, or from an activity standpoint, we're seeing what we're seeing, it doesn't change anything. It doesn't bring, what I'm trying to say is, it doesn't bring in that mechanistic or pathophysiologic piece.
I guess I was trying maybe to suggest more that are there ways you could fill in to try and explain this potential disconnect between how patients think they're doing and, and their actual activity is better, that is, or make patients realize they maybe, I don't know, but so maybe maybe what we should do, you're already thinking about other proposals. There are internal mechanisms of funding you could apply for as well. If you put together specific aims and a research strategy, 6 pages, for an RO3 type award, and let's just run it through a mock study section, and your external person that you got the suggestion from that it should be submitted to NIH. Let's get that person as a reviewer and a couple other experienced people in the field to do a mock NIH style review, and we'll see what they say. And then, nothing to lose with that way. I think I'd be really happy to try and get you some reviewers who can give you all that feedback, since we're interested, and I'm excited to see it move forward. But I don't think doesn't seem like anyone here has the full answer to the questions you're asking.
Yeah, but that is helpful, as in at least the thought process around trying to explain the discordance, the pathophysiologic bend or the physiologic bend to it. So I'll give that section a little bit more thought. At least that direction helps a lot.
Okay. Well, it is 1:31. Yeah, so please be in touch. Let's keep talking about this work. So let's
Absolutely.
switch gears to Dr. Tu's presentation, and we have a half an hour left. Yaping, I'll let you decide how you want to deal with that with your other collaborator, and whether we go past time. Thank you for being here.
Okay. So should I just start it? Can
Yep.
the screen
Oh,
sharing? Fine. Okay. So I'm the Dr. Chu from Department of Pharmacology and Neuroscience. So today, myself and my collaborator will talk about the new targeted therapy for severe and difficult treated lung disease, particularly like asthma and pulmonary fibrosis. So what is the asthma? It's a common chronic, inflammatory disease of the airways. And every year, there's worldwide, there's about 200 to 300 million people have the asthma. And every year, it's about a quarter of people died of this disease. And so what happened to the lung during the asthma attack? So this is a normal airway look like, and you had a smooth muscle, you had an airway. And during the asthma attack, the major change is that the airway become narrow. So there's the airway smooth muscle contracted and make the airway narrow. So this is one of the example. You show this is a mouse airway when they exposed to the bronchoconstrictors. So the airway will narrow, and which will create a significant increase to the airway resistance of that. The second one, so here's a slide show that how when the airway exposed to the different person, healthy person, mild asthma or severe asthma, when they expose the bronchoconstrictor, what airway constriction happened. And a healthy person does that do constrict, but here's the mild asthma, here's severe asthma. So significant increase the sensitivity of airway in asthma person. So we call the airway hyper responsiveness. The second called hallmark is called airway inflammation, and show we here. This is not only cause more mucus secretion, but also they change the airway structure and also create make the airway much narrowly. So this is therefore in clinical, the general treatment for the asthma is either increase the airway size called bronchodilation, or use the anti-inflammatory agents. So the bronchodilator most commonly used is called beta agonist. It could both the short-term and the long-term beta agonist. For the anti-inflammatory agent, there's drugs available, chronic use called corticosteroids. So this is and they inhaled or orally, and inhaled corticosteroids more commonly used that. And as a matter of fact, for most severe asthma, the typical use is a broncho, beta agonist plus the corticosteroid. But unfortunately, some patients not response to those drugs, and their asthma is poorly controlled, and they have more like at least twice the more like a severe asthma attack per year. So this is what we call this called a severe refractory asthma, or the severe and difficult treated asthma. So what really happened, and what is the underlying mechanism under this phenomenon? And we know that the beta agonist, why the beta agonist can induce bronchodilation, is that beta agonist will bind to the beta adrenergic receptor, and so the GS signal pathway, and this is G protein receptor, activate adenosine and then produce the cyclin-MP, and the PK will be active, eventually will trigger the relaxation of the airway smooth muscle. So this will relax, cause bronchodilation of the airway. But unfortunately, some of the patients show some kind of beta agonist insensitivity, in part due to the change of the beta adrenergic receptor, either mutations or because of the overuse of drugs cause the desensitization of the adrenergic receptor. That's the one of the issue. The second issue is that about inflammation. And for majority of asthma, the inflammation we call the T2 inflammation, and when those allergens or the pathogen trigger the inflammation, it causes the T2 inflammation secreted like TH4, interleukin 4, 13, and 5, eventually will recruit the eosinophil to the airway. And those patients typically is glucose sensitive, so they can be treated well managed by those glucose therapy. But some of the patients, instead of a T2, they call the non-T2 inflammation. What happens is that if instead of trigger the interleukin 4, 13, and 5, it goes to the interleukin 17 or 21 and 22. This is called Th17 dependent inflammation. And instead of recruit the eosinophil, they recruit the neutrophil to the airway. And the neutrophil mediated inflammation is insensitive corticosteroid treatment. So therefore, it forms as this corticosteroid insensitive asthma. Overall, this is only a small fraction of asthma patient, only about 5 to 10% of the patient. But however, it costs over 70% of cost, and also is a major cause of asthma patient death. And so far, there's no treatment available for this group of patients. So therefore, it's the significant lead for the development of the new treatment. And in the last 5 years, we collaborated with the different research group, both national or international, to screening, try to looking for the drugs can treat those, use the animal as the model to treat those refractory asthma. And one of the drugs we identify is the isolate from a Chinese herb called osto, and it form a complex with a phosphodiesterase, the block phosphodiesterase the function. And since the phosphodiesterase is an enzyme to degrade cyclin-MP, this one can trigger the airway, the bronchodilation, even when the asthma showed some kind of like a resistance to the beta agonist. So this is the one of the example. And the work was published as cover page as Science, Signaling, and about a year and a half ago. And furthermore, we first wanted to look for other, new bronchodilator, and so obviously we need to develop some kind of a system, the screening system. And this is one of the example. We call the precision cut lung slides, and so here's how this works. And you expose the airway of the lung slides and to the bronchoconstrictor. The airway will constrict, and then you treat those airway, constrict the airway with the different drugs, and some of drug will induce the bronchodilation as we show here. So there's a constriction happens, and then you treat it with the drugs induce bronchodilation. So this is and in addition, we generated called exception, in vitro model, vivo model that is called beta agonist insensitivity, the model. Basically, so that we treated this lung site with the albuterol, which is a beta-2 agonist for overnight, and that will cause the beta adrenergic receptor desensitization. And what happens that here's then you do the same performance just what we show in these videos. And what happens that here's the one control the tissue, and the albuterol induce relaxation. But when the albuterol pre-treated the lung site exposed to albuterol again, you see that not only they decrease the maximal relaxation, and also they shifted the dose to the right, they decrease the efficacy of the potency of the drugs. So we use this model to do some collaboration, through some collaboration with screening a drug compound library from Sigma, and this is one have over 1,000 different FDA-approved drugs or the pharmacologically active compound. And here, just one of the examples show that you pre-constrict the lung, and then you add those drugs and see how the constriction of lung decrease. And surprising, we find that one of drugs is called pirfenidone, and really very effectively induced bronchial dilation. Pirfenidone is the drugs, FDA-approved drugs, about 3 years ago. We used to treat the idiopathic pulmonary fibrosis, but even now the drugs are approved by FDA, but the mechanism and the target of the pirfenidone is still unknown. And in the last few years, we have been studying that how this pirfenidone works in lung. And quite interesting, this is a very simple small molecule, but what we show here is that we use the different the lung slides from the different species, from mouse, rat, or even human, and we show that they all show that it's very effective bronchial dilate, and it's about like from the EC50 is around 10 to 100 micromole, depending on what kind of the bronchial constrictors. We try the 4 different bronchial constrictors, show that the pirfenidone is a novel bronchial dilator. That's nobody know. And so after that, we obviously want to see how these drugs works. And the airway smoothable contraction is the one responsible for the airway constriction, and also is responsible for the airway hyperresponsiveness. So therefore, first thing we want to look at is that whether this is a pirfenidone really directly targeted the airway smooth muscle. So this is through the collaboration with the Dr. Stephen N. And they used this magnetic twisting cytometry, the method to measure the contraction and relaxation of airway smooth muscle cell. And here's I want just to show the data. And what we show here is that here's when you add the agonist, so the airway muscle contract, but when you pre-treated with the drugs or pirfenidone, they decrease the bronchial constriction induced the contraction. But not only that, if you pre-constrict the airway smooth muscle and then you add the pirfenidone, you see that there's a relaxation of the airway smooth muscle. So suggests that the pirfenidone can directly target the airway smooth muscle contraction and relaxation. And we have done a lot of work trying to search what target it is. And here's one of the example. So typically we think the bronchial constrictor, how to induce the airway smooth muscle contraction is that through the receptor, and then through the GQ and the PLC increase calcium in the smooth muscle, then trigger cause the myosin light chain protein to be phosphorated through this kinase, and that will induce contraction. But we also know now there's another pathway which is also G protein coupled receptor signaling pathway mediated, that is the Rho and Rho kinase ROCK dependent contraction, and which is block this MLC phosphatase, which is will further increase the bronchial constriction. And in asthmatic patient, this pathway have been known be amplified, but so far there's no drug available targeted this. Part of the reason is that the Rho and ROCK signaling pathway is so important in normal cell function. If you target this, the severe side effect will be observed. So instead of we target this Rho or ROCK signal pathway, in the last few years, we look for the upstream of the Rho signal pathway. That is the Rho GF. It's one mediate the signal pathway from G protein coupled receptor to a Rho. And here's just one of the example. We did a kind of a screening, for the 14 different Rho GF in compare the asthmatic and the non-asthmatic patient, and find out one of the Rho GF 25. And this one is compared to asthmatic was non-asthmatic airway smooth one. There's this one significant increase as compared to others. And also when we silence this Rho GF, and not only Rho activation was blocked, but also we find that this is also important for the function of the airway smooth muscle cell. Rho GF, this one is Rho GF is quite interesting because it's selectively be activated by GQ signaling pathway. And we know that GQ signaling pathway really mediated the contraction of airway smooth muscle. So therefore, this is could be a very important molecule to mediate, not only important for the GQ mediated, the PLC and the contractile pathway, but also mediate the Rho signaling pathway. So through collaboration with the Dr. Gopal Jagan, and he's the Malaysian chemist, and we find out that our perfidone fit into the this Rho binding site on this Rho GF, and based on this docking, and we also collaborate with my former postdoc, Dr. Shuni, and we synthesize a new compound called CXN8. And this one, through collaboration with Dr. Abel, we already received a patent for these drugs. And what we find out that these drugs, the CXN8, supposedly binds to the more potently, more tightly to this Rho GF. And from the study indeed show that it's really kind of like enhanced the bronchodilation effect. And we did also other things show that these drugs not only induced the relaxation of the airway smooth muscle cell and airway, but not only to the normal airway smooth muscle, but also in the smooth muscle cell, not insensitive to the beta adrenergic receptor agonist. So this has become very useful. So the bottom line is that this is a very potent bronchodilator as even in when there's a beta agonist insensitivity. Second one is what our data suggests that is really targeted the Rho GF and dependent Rho signaling pathway. So this is the initial. So the next thing, so what we did is that this is all in vitro study. So we did an in vivo study, used this mouse model. We called it a house dust mite model, and indeed we show that. So this one, when the mice exposed to house dust mite, not only they will trigger the airway inflammation, but also increase the airway hyperresponsiveness. And when we treat the mice with either perfidone or our drugs, which is 20 times less, and it works, and it reduced the airway resistance. And what quite interesting, this dexone, this is the corticosteroid steroid, it also works in partially, not as good as our drugs. And we know that this is dexone is a corticosteroid, right? So therefore, there's inflammation involved in that. As I said, there's two kinds of inflammation in asthma, either T2 or non-T2. And one is the eosinophil dependent, and another one is neutrophil dependent. But what we find out is that this HDM model is kind of like a mix of these two, both eosinophil and neutrophil. This is a very good model, which is mimic in clinical patient. That's partially is the neutrophil, partially eosinophil. Quite interesting is that when we use the drug to this, what we find out that dexone have no effect on the neutrophil, the inflammation. And this here's the Dexon, here's the vehicle. But both the pirfenidone and CXN8 can reduce the neutrophil, the mediated inflammation. Of course, the eosinophil mediated inflammation can be blocked by all three. So we did so therefore we generated another model that is a overbumin LPS model, which is more like show that it's 80% of the inflammation cell in the lavage is all like it's neutrophil. And what we find out our drugs also works, but not the Dexon. So this data suggests that our drugs is also not only block eosinophil mediated inflammation, but also block the neutrophil mediated inflammation. And we have the data show that this is the Th17 dependent response, because in vitro our drugs, but not Dexon, can block the T17 dependent inflammation and in vitro. So overall, here's our overall hypothesis. That is, for the refractory asthma, there's two called hall marker. One is the airway hyperresponse due to the airway smooth hypercontractivity. Another one is a neutrophilic airway inflammation due to the Th17 dependent inflammation. And what we show here, our drugs can block the function of this P60 ROGF, and this molecule play a very important role to stimulate the ROG signal pathway that is important for both the airway smooth muscle hypercontractivity and our Th17 differentiation. So we have submitted a grant to the NIH, and the first submission we got a 20 percentile. So we're planning to resubmit it back to this fall. So I think I'm gonna stop here. So a little bit over. Okay.
Really, really exciting work, Yaping. I wonder about the dose of the the pirfenidone in your mouse study versus like its human use in the IPF.
Yeah, the drugs, the pirfenidone use the drugs dose is, and since mouse is metabolizes is faster than so we have about 3 times than human, but that's the typical 300 milligram orally form is the one. And when we do the animal study, the people show in the animal study of the pirfenidone. And what I want to say is that in fact in here we collaborate with the Chris now school. So we are going to develop some kind of a microparticle loaded with these drugs, and the preliminary data show that it will significantly change the pharmacokinetics and increase their half-life time by 10-fold. So therefore, we can decrease at least the dose by 10-fold.
Seems like it would be great to try the pirfenidone in an off-label study, or look in the literature or study the IPF patients. I don't know if any of them have asthma, but like to try and get that drug. We are trial for asthma.
We are currently collaborating with Dr. Tom Casati. He's the former chief the immunologist in our school. So they moved to University of South Florida. But I will be happy to have some kind of a clinical people because this pirfenidone is already proved, and the dose we are using is, as a matter of fact, I think that we can use low dose because at least in fibrosis, we also studied the fibrosis effect, and typically for the fibrosis in vitro, the EC50 is about 1 millimolar, and what we show that the pirfenidone is around 100 micromolar. So therefore, I feel like in the clinic side we can use low dose for treat those asthma patients.
Yeah. Any pulmonologists interested?
Yeah, I have a collaborator, so I think so. Next step, we're currently doing the collaboration on the pulmonary fibrosis, but I think the next step which hopefully we first get this RB approved, then we'll add more sample for the asthma research.
It's beautiful work. Anybody else? I feel like I'm monopolizing the conversation.
If not, If no question,
It's no, Maureen
okay.
Tierney. I just wanted to echo that I
Okay.
think this is really exciting work. And I think, that sort of clinically how we see, like in so many conditions, but there's the group of people who are most effective are you often different in some way from the folks who are affected in a moderate way. So to see that there's, to see your work kind of explaining why that is really enlightening from a clinical standpoint. And I could see, a number of pulmonary docs potentially being interested to collaborate with you when you're ready at the next step.
Yeah, that, as a matter of fact, initially we want to just look at a new bronchodilator, and then we did a mouse study, and the data was striking. Not only blocked eosinophil inflammation, which is typical that way, but then we saw those neutrophil inflammation.
Yeah.
And the good things that we use the right model. This is a HDM model. If you over-human model, it's only the eosinophil. It's a mix of the eosinophil and the neutrophil, and we see the neutrophil, the inflammation totally blocked, and the dextron have no effect on the eosinophil. So that makes us to think about the potential application even stronger. Not only just as a bronchodilator, but also they can block the inflammation. I should point out is that when this drug pirfenidone was initially designed, it's designed as the anti-inflammation drugs, and then they later find out it's treated for pulmonary fibrosis, but they don't know the target. I think we have data show that low signal paths are also important for the fibroblast, the differentiation. So therefore, I think this may be the one of the target underlying why the pirfenidone works for the pulmonary fibrosis patient.
Can I ask one other question? And if you may have mentioned this and I missed it, but how do you make sure that the folks where there's a lot of neutrophil activation don't potentially have a bacterial sort of superinfection that's activating neutrophils? Is that part of your process?
I think part of it, what I talked to the Dr. Casati. He think that part of the neutrophil inflammation is caused by those bacterial infection, and there's a bacteria we wrote a review recently. I think a bacteria it's part of the reason, and in fact, the one of a mouse model were used for those neutrophil asthma model is used LPS, which is come from the bacteria. I think that's the part of the reason why those patient is not very sensitive to is due to the bacterial infection in the airway.
Thank you.
Okay, I think I'm gonna transfer to my collaborator.
Thanks so much, Dr. Jagan and Dr. Tu, both of you for presenting, and everybody for being here. And we're recording the session, so I will come back and watch this third presentation, but go ahead and carry it away.
Yeah, sure. Can you just start sharing your screen?
Sure. Are you able to see it now?
Yes.
All right. Hi, everyone. Thank you for having me here. I know we're a little short on time, but this is a very brief presentation. So I'm Shraddha Narachania. I'm one of the attendings here in the pulmonary department, so on the clinical side. And I'm happy to collaborate with Dr. Tu on this project. So again, it's just going to be 5 or 6 slides. I'll try to keep it brief and very short. So to follow up on what Dr. Tu just presented here about his research on asthma. So there was another paper that Dr. Tu had published with his colleagues in 2016, which was this one, Upregulation of RGS2, a new mechanism for pirfenidone amelioration of pulmonary fibrosis. So this was just, looking at a novel biomarker in patients with IPF. There's many out there. This is potentially another one. So what Dr. Tu's team did is they took human lung fibroblast cells. They exposed those cells to pirfenidone. Pirfenidone is, as he had mentioned before, it's an anti-fibrotic agent, and it's currently FDA approved for patients who have IPF. He exposed these cells to pirfenidone, and then they used gene chip microarray to screen for genes that were upregulated by the presence of pirfenidone. And they found this one gene called RGS2, which is regulator of G protein signaling 2, which was one of the genes that was upregulated by pirfenidone. So what they also found is that when they took bleomycin-induced, the rat model, bleomycin-induced pulmonary fibrosis, pirfenidone actually reduced bleomycin-induced fibrosis in the wild-type mice that had the RGS2 genes, but it did not do that in the RGS2 knockout mice. And again, I've just very briefly summarized this. I'm not expanding too much on what they found and the details, but what they concluded from the study is that endogenous RGS2 does exhibit anti-fibrotic functions. And when this particular gene is upregulated, that could contribute significantly to the anti-fibrotic effects of the drug pirfenidone. So based on this study, we came up with a hypothesis for our own study, and we're collaborating on that currently. So this is kind of a pilot study, but our hypothesis is that in patients that have idiopathic pulmonary fibrosis, they have reduced expression of this gene, RGS2, and they also have increase in RGS2 gene polymorphisms in their peripheral blood mononuclear cells. So what we are planning to do is have two groups. We're going to have 20 patients in each group, again, because it's a pilot study. But we're going to have an IPF group and a healthy volunteer group. The IPF patients are going to be recruited from our interstitial lung disease clinic here in Bergen-Mercy. We're going to be taking adult patients 19 years or older who have IPF diagnosed for the ATS criteria, which is American Thoracic Society 2018 criteria. We are going to be excluding patients that have a current or a past history of smoking or vaping. And the reason for that is because smoking has actually been shown in animal models to also affect RGS2 gene expression. I think it downregulates it. So we're trying to avoid that. And pregnant patients. And then as far as the healthy volunteer group, these patients are actually going to be recruited from our sleep clinic. So the sleep clinic, usually sees a lot of patients that have obstructive sleep apnea. So we're going to try to get those patients, but we're going to make sure that they don't have any kind of chronic lung disease or any chronic respiratory issues. Again, there'll be adult patients. We'll try to match them to our IPF group. And then we'll be excluding, again, patients that have a history of smoking, vaping, pregnant patients, and as I mentioned, anyone with any kind of chronic respiratory issues or lower respiratory issues. I mentioned where these patients will be enrolled from, and then once we enroll these patients, we're going to be collecting their blood. It's about 15 cc's of blood that we'll be getting from each patient. And once it's collected, it's going to be sent to Dr. Abel and Dr. Tu's lab at Creighton. And Dr. Tu is then going to be isolating the mononuclear cells from this blood. And he's going to be then looking at their DNA, RNA, and looking at the expression of this particular gene, RGS2. That's about all we have right now. That's what we're planning to do. This is in the IRB approval process, and we're hoping to get it soon. And once it's done, we're hoping to start recruiting patients in this. So that's all I had. Thank you so much, guys, for having me here. And I'm open to any questions, any suggestions that anyone may have.
So I had a question. The questions that, I mean, this one I just choose quite interesting because it functions as a negative regulator of GQ signal pathway.
Yes.
And we previously showed that this GQ signal pathway, it's important for asthma. Then we showed that this is one of gene. And in fact, we and Clary just published to show that this gene is polymorphism. One of the polymorphisms we identify is associated with the severe asthma. So I think they will be quite interesting to see whether this is in the IPF patient, there's any genetic polymorphism happen to that. Not only that, I would like, once this IRB approved, I would like to add another group as like either severe asthma patient, as you still have a control group there. So we do some kind of comparison. That would be, instead of now just two groups, but add a third group, that will be interesting to see whether there's anything, any change. And in the future, and would be great to have another group which is treated with the pirfenidone, asthmatic patient with pirfenidone treated group. The fourth group will be provided even more important information to this gene. Yeah.
Right. And I think that's part of our questionnaire for these IPF patients as well, because I think we've included whether or not they are on antifibrotic drugs as part of the questionnaire. So I think we'll be able to get some data on that, hopefully, because most of these patients will usually be on some kind, either it'll be pirfenidone or the nintedanib, which is the other medication that's very commonly used. So.
Yeah, great study, Dr. Narachania. This is Gary and Curry. So in your questionnaire, did you include if they had childhood asthma?
No, so I don't think we've included that. This was only meant for IPF. But I think if they have any history of asthma, I think we're going to be excluding these patients. I think it's just going to be purely IPF, no other respiratory issues other than IPF.
Even in the past, that is. So no history of, okay.
As of now, I think we, myself, and I think we, like our clinical team talked with Dr. Abel and Dr. Tu initially when we were trying to get the design. And I think we just wanted it to be a clean study for now and not have any confounders. So.
Agreed. Yeah, no, I was just wondering because he just mentioned asthma. So I was just wondering if childhood asthma,
yes,
just to see if there, you may get a subgroup analysis there.
Right, right. I think so. The hope is, I think if we get any kind of signal from this study, from these 40 patients, then I think you could make it into a prospective study and enroll more patients.
Yeah, the good things are that we have this homozygote and heterozygote knockout mice. So anything once we find out there's any differences, so we can use those mice to mimic that and see whether there's how this is this gene play a role when they're downregulated.
Right.
Okay.
Fascinating stuff. Thank you.
Thank you so much.
Okay. Justin?
Yeah, does anybody have any additional comments or questions or any input? If not, I think Dr. Hansen has stepped out of the room. So I think we can go ahead and end. Thanks everybody for all the input on these projects. It was a lot of fun to listen to. I believe there will be some information sent out about the next research brainstorming session in the days to come. So keep your eyes peeled for that. And thanks again.
Thank you, Justin. Thank you for attending.
Thank you, everyone.
Thank you all.
Thank you. It was wonderful listening to these two presentations, and I think it's a great model for future collaborations with other basic scientists and clinicians. So thank you very much.
Okay.